When we place a microscopic organism in a highly controlled laboratory environment, we can force it to reveal its genetic secrets. We watch it divide, mutate, and multiply exponentially to fill its vessel. This is the modern framework of evolve and resequence, or ENR, studies. By combining strict evolutionary pressures with next-generation sequencing, we track the genetic composition of an entire population over time. The foundational premise relies on a simple observation. If a specific mutation rapidly rises to high frequency under our controlled conditions, it is highly likely to be an adaptive driver mutation. However, a severe biological obstacle corrupts this clean premise. That obstacle is linkage disequilibrium. When an adaptive driver mutation arises on a chromosome, it does not exist in isolation. It is surrounded by neutral, neighboring mutations. Because these adjacent nucleotides are physically tethered to the driver on the same strand of DNA, they get dragged along for the ride, a phenomenon known as the hitchhiking effect. As the driver mutation sweeps through the population, the neutral passenger mutations sweep right alongside it. This creates a massive noise floor in our sequencing data. You are left staring at thousands of high-frequency mutations, making it nearly impossible to isolate the true target of selection. To solve this, we have to systematically optimize the core parameters of our experimental design matrix: population size, replication, and duration. By tuning these variables, we can break genetic linkage. By utilizing robust forward simulation genomic data, we can define the exact parameters required to filter out the hitchhikers. Coordinating these three parameters yields a high-throughput screen for the molecular targets of selection. To distinguish one causal locus from millions of neutrally evolving ones, researchers rely on statistical tests that contrast allele frequencies between the base population and the evolved population. This graph plots the ultimate metric of success for these statistical tests, the true positive rate on the y-axis against the false positive rate on the x-axis. A poor experimental design yields the baseline curve shown here, catching too many false positives. Our efficacy depends strictly on pushing this signal-to-noise curve upward and left, maximizing true discoveries while suppressing false ones. Experimenters manipulate a three-column design matrix to achieve this ideal curve: duration measured in generations, the effective population size, and the number of biological replicates. Finding the causal loci is a direct function of how successfully we tune this matrix to strip away neutral background noise. We begin with the first tunable parameter: duration, measured strictly by the number of generations. Simulation data yields a clear directive. Extending an experiment from 60 to 300 generations significantly increases the discovery of true positives. The biological mechanism driving this increase is time-dependent recombination. More generations directly equate to more crossover events occurring during reproduction. With every crossover, neutral passenger alleles are systematically sheared away and moved on to unselected haplotypes, effectively isolating the driver mutation. There is a crucial nuance to this metric. The duration effect is significantly more pronounced for weakly selected loci than for strongly selected ones. Strong selection is efficient. It drives advantageous haplotypes to fixation rapidly, closing the window of time required for recombination to act. Conversely, weakly selected alleles take far longer to sweep through a population. This prolonged timeline gives recombination the opportunity to break the linkage disequilibrium. Because of this dynamic, short-duration experiments are heavily biased. They primarily detect major effect mutations, while missing subtle genetic shifts.
Evolutionary time provides the mechanical shearing to strip away hitchhikers. Long durations are necessary for discovering the subtle, weak adaptations that define most evolutionary change. The second parameter of the design matrix is the effective population size. Maintaining massive population sizes, thousands for insects, millions for microbes, is a requirement in experimental evolution. A large population fulfills two primary functions. First, it harbors a reservoir of initial standing genetic variation, providing the raw material for adaptation to occur. Second, it mathematically overpowers genetic drift. These two side-by-side charts track allele frequency trajectories over time in a small population versus a large one. In the small population pool, stochastic drift acts as a chaotic force. Random, unselected alleles wildly fluctuate in frequency and can accidentally reach fixation. In the large population pool, sheer numbers dampen those random fluctuations, stabilizing the background noise. This relationship is governed by a strict mathematical inequality. The product of effective population size and selection strength must be vastly greater than 1. When an experiment meets this threshold, deterministic natural selection becomes the dominant force dictating survival in the laboratory vessel. Without massive population sizes, stochastic drift acts as blinding statistical noise, entirely obscuring the deterministic signal of beneficial mutations. The final parameter in the E&R arsenal is biological replication. Researchers frequently face a resource dilemma with finite budget and incubator space. Should they prioritize maximizing the size of a single population or dividing those individuals into multiple replicates? The simulation data provides a clear mandate. You must prioritize replicates. We are looking at a split-screen comparison to understand why. Each replicate acts as a completely independent recombination vessel. In a one-replicate setup, a neutral passenger mutation can easily get lucky, hitchhike alongside a driver, and permanently corrupt the data. You cannot distinguish the driver from the passenger. In a 10-replicate setup, the passenger mutations that randomly hitchhike in replicate A will be completely different from those in replicate B. The noise is localized and random. While that noise varies wildly, the true adaptive driver will consistently spike across every single independent population, separating itself from the background. Biological replication is the mathematical filter used to cancel out the localized random noise of linkage disequilibrium across an entire study. When we synthesize these 3 parameters, we generate an actionable laboratory blueprint. These 2 Manhattan plots show the resulting genomic peaks from 2 extremes of experimental design, a compromised setup versus a fully optimized one. The low-budget matrix is restricted. It relies on 3 replicates, a small population of 500, and a short duration of 60 generations. The resulting genomic data is riddled with messy, numerous peaks. The researcher is blinded by false positives, and discovery is restricted only to major mutations. The high-budget, optimized matrix expands to 10 replicates, a large effective population of 2,000, and pushes the duration to 120 generations. Here, the false positives drop away. The noise floor collapses, leaving only sharp, isolated green peaks of the true selected loci. By utilizing this optimized matrix, researchers can accurately map up to 56% of all selected loci, down to single nucleotide resolution. This architecture provides the sensitivity to detect selective advantages as low as S equals 0.005. Standardizing population size, replication, and time according to these models allows researchers to systematically identify the genetic variations that drive survival.